Hello everybody. I am Dr. Farhan Zamir, an adjunct professor and academic specialist at Biotechnica, Bangalore. Today we have an important topic for our discussion, and the important topic is what is the importance of R language in bioinformatics. So to understand this, let's dive in. Now, as I have discussed, what exactly is the importance of R? Why should I learn R? Now, if you look into the recent trends of programming or computing languages, so you see most of them who have learned C++, Java or Perl or Python are now just transcending and using R for their analysis. So this is much more evident if you read certain recently published papers wherein you have beautiful plots, beautiful diagrams, and the data interpretation is simply amazing. Now, with that, we thought, let us try to see what would be the importance of R language in bioinformatics. Now, remember, R as a language was developed by statisticians for statisticians. So it was never meant for bioinformatics. But however, there are a lot of tools which were actually designed just for executing statistical problems. Now, one such tool is SAS, which is suite for analytical software, or it is also, you have a different packaging called as SPSS, which is statistical package for social sciences. You have something called as Tableau, you have the Alteryx, you have Prism GraphPad, Origin, and very simple, you have Excel Stat from Microsoft. Now, all these are statistical tools which are generally used for analyzing demographic data or a data which can be looked into a log graph or a semi-log graph, okay, or something which we are trying to analyze a business of a company, income, expenditure, all those things. When it is on a huge number, people are trying to look upon or develop new strategies in their business component. But however, it was somewhere in 1980 wherein John Chambers, who actually designed a language which was called as S language. This S language, as it was developed from the Bell Laboratories, later it was somewhere in 1980s or 88 or 1990s, it was Robert Gentleman and Ross Hucker who were able to come up with a derivative of S language, and since their name starts with RR, the program was called as R program or R language. Now here, what are the advantages of R? Now please trust me, my dear friends, R is absolutely free and it is an open source in nature. So this means to say I can just go get into an R console or an R terminal and just download it for free. Now, you need to have a prerequisite of a good RAM, a good computer, so for downloading all these things. Otherwise, and in between the program would just get stuck. So the console is a bit bigger so that you need to actually have a proper computational technical tools for installing R. Now, R is also hugely on statistical computing, design, and also it is predominantly was meant for giving up plots and graphs with various annotations and various peak symmetry. Now, it was also very easy to understand since the syntax which has been used is very simple, and also the interface which is being used is great. So you can always have an R with an hybrid language system wherein you can amalgamate R with that of Perl, R with Java, R with Python, and that is how there is an emergence of new languages which is called as hybrid languages, which we have spoken in other video. Now, concluding this particular slide, what we are trying to understand with R Now, the trend of R is simply making R as the language for data sciences. Now, trust me, my dear friends, in this era of data revolution, everything is data. Now, for all this analysis, might be statistics or might be biological data, R plays a very important role, and hence we conclude R as the language of data sciences. Now, with this R, there comes an additional add-on. There's an additional piece of software which is much more compatible to that of R, and this is called as R Studio. Now, this R Studio has a lot of advantage. The advantage is it is having a consistent commands, and it is comes under with a unified window. Now, anybody who has already previously used R, you can see there are multiple windows, mainly there are 4 to 5 windows which actually pops in. Now, this becomes very troublesome when you're actually reading this data. But however, RStudio gives you one unified window which allows you to input or execute the data, and that is a beautiful advantage of R. Now, this also helps in optimizing your vector operations. And very importantly, in R, you can have a free navigation. So, especially in terms of your table or in your loop, so R or RStudio gives you an option to navigate. And not to forget, it has a great community. So, all renowned coders who are actually the inventors of it, and also people, as the R program progressed, there are a lot of commands which have been deposited, and this any point of time is a great community. And if you are stuck somewhere with your programming or with your coding in terms of R language, this community comes to your rescue. So it gives you a lot of add-on advantage to have an interaction with this community. Now, also not to forget, RStudio gives you an add-on advantage of around 9,000 plus packages which could be an integral component of R, and then these 9,000 packages also involves a lot of softwares, a lot of tools which could be used for bioinformatics. There is something called as Cranks. This is a comprehensive R archive network. This Cranks helps us in understanding a huge component of data, especially coming out of biology, which has a beautiful interpretation tools and plotting tools. So we'll try to look into all these things as we see what are the advantages or what are the applications of R, especially in terms of bioinformatics. Now, before I go into the bioinformatic component of it, R is now basically used for understanding biostatistics. Since in the beginning of the video, I told you it was a program which was generated or created by the statisticians to the statisticians. So here, in terms of biostatistics, a lot of biostatistic data is now being analyzed with R program. So it gives you a comparative analysis. And also, very importantly, looking into the significance level of your data. Depending on that, you can create your p-value clusters, or you can create your p-value loops, look for the upregulation and downregulation of the data, and very importantly, validation and interpretation of the data, especially in biostatistics, has become very, very easy because of the application of R. Now, in bioinformatics, R is mainly used for protein structure. Now, in protein structure, R could be used for aligning sequences. Now, trust me, we normally use an NCBI tool which is called as BLAST. BLAST refers to Basic Local Alignment Search Tool. However, R also does the same, but however, they have a completely different kind of a BLAST which is called as R-BLAST, which will also help in not just aligning sequences, it will help in aligning genomes. Yes, you heard me right. It will allow to align genomes. So one complete genome of an elephant could be mapped against a genome of an ant, and this is a powerful tool which has been called as R-BLAST. Now, this also helps in understanding the protein structure prediction, and very importantly, the molecular dynamics and stimulation studies with their validation could be performed using R language. Another important application of R would be in DNA sequencing. Now, when we sequence a DNA, so a DNA, many a times when you sequence, there are two major components. One is a huge chunk of sequences which are absolutely no use for me, and many a times there is a very small set of sequences which are very much useful for me. Now, this are being mixed in a complete big stack, and removing this important data, that is removing a novel data from this garbage, becomes very, very difficult. Now, manually, I tell you, my dear friends, it is impossible to do a manual curation on a DNA sequence. So what we do is we use R programming to clean up the data. Many a times the sequencing data is having a lot of adapters and chimeras. This could be easily chopped up using R program. Aligning the context becomes very, very easy with R. Validation of context and validation of OTU. OTUs are operational taxonomical units, becomes much more easier. And once I have the validated context, what I can do is I can simply go into a plot analysis, and this is called as ggplots in R, and which will give you fantastic curves which are much more readable and much more understandable when you are actually putting that data into your manuscripts or into your research papers. Now, as I was trying to make you understand that the application of R could be in a huge component of your RNA or your array data. Now, it could be either an RNA array, it could be a DNA array or a protein array. So any of these arrays will actually have a huge amount of data. Analyzing this using R actually becomes very, very easy. Now, this will help us in understanding the regulatory cascades. This will also make us understand, the upregulation or downregulation of the genes. And very importantly, if there are certain set of genes which are neutral genes or which are totally not related genes which are not responding for your treatment, so these particular genes could be actually excluded from your data, and then the highly expressed genes, either based on the p-value or based on their scientific significance, could be analyzed with their fold ratio, and this expression and validation of the data becomes very, very simple using R array data. Now, also R plays a very important role in developing phylogeny. Now, what is phylogeny? You are trying to develop an ancestral character, ancestral feature. Say, for example, if you want to analyze keratin. Now, keratin was present in Ramapithecus, Australopithecus, Cryopithecus, Bryopithecus, and now Homo sapiens. Now, you want to do a phylogeny. At what point of time did the cilia, did the hair become fur? And from fur, it again came back into cilia. So, if you want to do this kind of an ancestral studies, I think R is one best option wherein you can actually analyze the genomes of various organisms and then try to develop a phylogenetic tree. So, helps in analyzing a variable data, and very importantly, loop parameters are being possible using R, and the tree representation which actually comes out of R is very, very readable, and this many a times could be either based on the bootstrap value variations, and the plots are very, very visible many a times on a free software. Okay, it is more of a crowded thing, but however, R, though it is free, it has a beautiful readable data. Now, the analysis of nodes and internodes becomes very, very easy when it is being analyzed during using R program, and phylogeny, understanding of phylogeny becomes very, very simple. Now, another important application is the application of R in network analysis. Now, in network analysis, we need to understand what is the association between one gene and another gene, or a gene or a protein, or a gene with an RNA. So, to look into all these kind of pattern studies or cluster studies, R plays a very, very important role. The second thing is to understand the association studies and draw the string networks through the association and integration studies. This becomes very, very easy with R, and as I previously mentioned, that the plots and graphs which comes out of R is simply fabulous. Now, The network analysis, normally we do it using string database, but however, or cytoscape, or however, when the data is much more huge, okay, string database has its own drawbacks, or cytoscape has its own drawbacks. Now R gives a comprehensive analysis of network, and this gives you a beautiful readable network which could be deposited in certain metabolic databases such as KEGG. Now, application of R in RNA-seq data plays a very, very important role. Since you know, RNA-seq data is a huge sequence of data of RNA, and there cleaning up of the data becomes very, very important. So once you clean the data, the fold expression has been done using R. Again, network analysis could be done, and then the pathway development through RNA-seq becomes very, very important, and so that I can extrapolate the data what I get from my dry lab into my wet lab studies. So R has a very important component in your RNA-seq data. So these were the few applications which was been mentioned, and the last and very important application is on your microbiome analysis. Now on microbiome analysis, as you know, if I take up a skin microbiome or a gut microbiome or any kind of a tissue microbiome, you have huge number of the phyla or the species of microorganisms which are present. Now understanding the entire sequences is simply impossible. Now R gives you an edge on it so that you can clean up the data, you can actually select a certain OTUs. So here you can take up operational taxonomical units and then coincide that with a specific sequence of your 16 years so that you can identify the microorganism or the microbiome which is of your interest. Then once you understand this, you can also cluster the microorganisms. Say for example, if you're looking for good bacteria and bad bacteria. Now when you look for good bacteria, many a times if you want to consider me as healthy, so then the concentration of the good bacteria should be higher and the bad bacteria should be lower. Now any point of time when the bad bacteria concentration increases, this means to say I'm transforming from a healthy individual to a diseased individual. So what can I do? I can identify these particular bad bacteria and utilize them as a biomarkers or identifying or as a diagnostic markers for the identification of that particular disorder or disease. And hence this kind of a research is actually going into oncology, wherein people are trying to design specific microbiome either for the treatment or for diagnosis. Now understanding the cluster analysis using PCO curves becomes again very important. Principal coordinate analysis plays a very, very important role when you take up a demographic data. So when you have a huge population, you are doing across demography, across culture, across races, across species, then this kind of an analysis has been properly plotted using R as a program. And very importantly, the validation and interpretation of microbiome data is simply great using R program. So as I was trying to tell you, R program also gives you an edge for designing hybrid languages, wherein you can amalgamate Perl with R, Python with R, Java with R, and then execute it for the much more readable and understandable data. So this was an effort to make you understand what exactly is the applications of R in bioinformatics. Now this story does not stop here. People have gone one step forward in applying R into artificial intelligence, machine learning, and deep learning systems, wherein the attributes are being learned using vector analysis. Annotation becomes much more easier in AI when R is implemented on it. There's something called as object-oriented programming, a beautiful and booming program, which is a program for future. So everything maybe down the lane in next 10 years, everything would be AI based or ML based. So hence, studying this language would be much more helpful. And very importantly, when we have something called as big data, so the big data analytics, you have lot of graphs, lot of interpretations which are involved, which will allow you to plot graphs, and very importantly, develop strategies, develop models which can actually fit into your biological system. So the entire intention of all these is to create something which is called as human biomimic. So people, they just want to do analysis on a chip which can mimic a complete human, and try it out. There is a beautiful branch of science which is called as organ on a chip, or a human on a chip. So this will again enable you people to understand the application of R in AI, in ML, or in deep learning, wherein we are simply trying to create human biomimics. So with this, I thank you people for sparing your time, and also very importantly, I request you people to go and check out with R. It is free, and maybe in initial stages you have certain hiccups, but however, once you understand the program, this is much more easier to get executed, and you can learn biology in a much better way. So at Biotecnika, our intention is your success is our priority. With this mantra, okay, I leave you and wish you all the best in learning R. Thank you very much. So if you have any comments or suggestions, please do not forget to write up a comment, and do not forget to subscribe. Thank you very much.